Hello, everyone. Good afternoon. My name is Tuli Racer, and I am the project manager of the IAS Corporate Partnership Program. And it is my pleasure to welcome you to this HIV vaccine industry partnership workshop on biomarkers and clinical trial design. This workshop, conducted in partnership with the Global HIV Vaccine Enterprise, follows from the series of events on design approaches for current and future HIV prevention efficacy trials. As a reminder, there are a couple of housekeeping rules for this workshop. The workshop will be recorded, and the recording will be made available to all participants in the coming weeks, together with a report. By staying in this meeting, you essentially consent to being recorded during the workshop. We encourage everyone to send questions to our presenters at any time throughout the workshop, either via the chat function or by raising your hand. And please do remember to focus on today's workshop theme when posting your questions. I would now like to hand over to the HIV Vaccine Industry Partnership Co-Chair, Linda Gale Becker, for a brief introduction to today's workshop. Over to you, Linda.
Thank you very much, Tuli, and my warm welcome to everyone who's joined us this evening, and hopefully will continue today and will continue to join us. It's evening for me here in Cape Town. I am the Executive Director of the Desmond Tutu HIV Centre based at the University of Cape Town here in Cape Town, and I'm a past president of the International AIDS Society. So this workshop brings together industry and non-industry representatives who have a stake in HIV vaccine R&D. And the idea today is to explore how biomarkers could be used as surrogate markers of clinical efficacy in counterfactual vaccine trials. Of course, it is an important time. I think it's been described as a prevention revolution. We've seen the onset of very exciting new prevention technology and biomedical interventions, particularly in the ARV-based prevention field. We also know that the broadly neutralizing antibody being infused as passive immunization is another important aspect of prevention potentially in the future. And whilst this is very exciting and certainly welcome for those of us who have grappled with seeing the end of the HIV epidemic, it also brings complexity and conundrums as to how we will in fact take the vaccine field forward. And particularly that pertains to efficacy trials and how efficacy trials will be feasible and affordable in the future. Because we know that placebo-controlled trials are probably something of the past. And this is really where the concept of counterfactual approaches, which may allow interesting other ways to evaluate product efficacy, come to the fore. And in particular, biomarkers as surrogate endpoints of vaccine efficacy are an approach that I think warrants evaluation. And if one or more biomarkers could reliably predict the efficacy of a vaccine in a clinical trial, thereby replacing the need for HIV infection as primary clinical endpoint, such markers obviously could then facilitate and accelerate and, of course, make affordable a research in this way from an early stage. So the use of biomarkers as surrogate endpoints for clinical efficacy in their own right come with a lot of challenges and limitations. These include identifying, validating, really, even as we realized last week at the CAVD meeting, even convincing many parts of the field that this is a way to go in itself is a challenge. So regulatory, scientific, as well as community perspectives also matter here. And today we'll be hopefully grappling with all of these sectors as we unpack this very important, I think, subject for the future of HIV prevention. So I am now very pleased to hand over to my co-chair, Carrie Wang, who joins me to help moderate this webinar today. Over to you, Carrie.
Great. Thank you, Linda Gale. And good morning, good afternoon, and good evening to all the participants for this meeting today. My name is Carrie Wong, and I serve as Senior Vice President in Clinical Research and Head of Chronic Infections at Vir Biotechnology. It is a pleasure to be here today to explore and discuss opportunities and challenges when it comes to using biomarkers as surrogate endpoints, and to debate the design of future efficacy trials. Following up on some of the points that Linda Gale made, conducting vaccine efficacy trials are perceived by many as a major hurdle by industry for some of the reasons that she listed. Several vaccine candidates are being developed, and we need to be able to evaluate them quickly in a cost-effective fashion to determine if they elicit immune responses and are able to prevent infection. What are these correlates of protection that we're looking for? Biomarkers of efficacy could also play an important role in testing vaccine candidates. And this is also true in cure research, where vaccines may also play a key role. So the IAS industry towards a HIV cure collaborative group recently held a workshop on this topic. And today we will be focusing more on preventive vaccines. So why do we need to, why is this so important? There remains a great unmet need in preventing new HIV infections. Around 1.5 million people became newly infected with HIV in 2021, with women and girls accounting for about 49% of all these new infections. Although new infections have declined by about 32% since 2010, we need an HIV vaccine for long-term control of HIV. And a simple to use vaccine would be an essential tool in reaching populations most affected by HIV. And a vaccine with even 50% efficacy would have a major impact on an epidemic. So today is a great opportunity for industry to engage with academic researchers in the community, ask questions, and make suggestions on how to develop these relevant and acceptable biomarkers, especially with regulatory approval in mind. So with that, I'll turn it back over to Tuli.
Thank you, Linda Gale and Carrie. This workshop will be in two parts with a break halfway through. In part one, we will have two presentations followed by a short Q&A session, after which we will have a group discussion. And the second part will include two presentations followed also by a Q&A session, after which we will dive into another discussion. We have a rather packed agenda today, so we ask all the speakers to please stick to your time slot in order to allow for more time for a discussion. And with that, we will start with a short introductory video on biomarkers and surrogate endpoints, followed by a recorded presentation by Sayan Dasgupta on evaluating the surrogacy of multiple vaccine-induced immune response biomarkers in HIV vaccine trials.
Hello, I'm Dr. Susan McCune. In this module, I'll discuss some ways that biomarkers are being used generally to improve drug development. Biomarkers are playing an increasingly important role in drug development. They can help improve clinical trial efficacy and reduce uncertainty in regulatory decision-making. But before we look at how biomarkers are improving drug development, it's worth noting some of the ways biomarkers are used in other scientific settings. Clinical biomarkers are used every day in healthcare. For example, some people track their blood pressure, a biomarker for cardiovascular health. Other people test their blood glucose levels as a biomarker to make sure their diabetes is under control. Biomarkers improve health through novel means of diagnosing disease and providing ways to determine responses to new treatments. Biomarkers have the potential to enhance public health through a better understanding of diseases and treatments. In addition to their use in everyday healthcare, biomarkers are important in the drug development paradigm. Drug development is really a continuum, starting with basic research and extending through drug discovery, preclinical development, clinical development, and post-market safety assessments. Biomarkers are routinely used across this continuum in studies to support drug approvals. For example, biomarkers can help to predict patients who might respond better to a drug from a safety or efficacy perspective, monitor the safety of a therapy, or determine if a treatment is having the desired effect on the body. Biomarker-based strategies allow for a more biology-targeted approach to drug development and may enable time and cost savings through leaner, more focused clinical trials that have a higher overall probability of success with respect to both efficacy and safety. Let's take a closer look at the role biomarkers play at different points in the drug development continuum. In the early stages of drug development and preclinical testing, biomarkers may help to identify molecular pathways that contribute to disease. During prototype design, biomarkers can be used to identify the mechanism of action of a drug or help in selection of the drug target. In the preclinical development phase, biomarkers can be used in preclinical safety assessments in identifying the mechanism of action of the drug or in helping with dose selection. Biomarkers can serve a wide range of functions in clinical trials, including stratification, patient selection, dose selection, trial enrichment, safety assessments, and efficacy assessments. Here are some examples. As an example of patient selection, galactomannan may be used as a diagnostic biomarker to classify patients as having probable invasive aspergillosis for enrollment into clinical trials of antifungal agents to treat invasive aspergillosis. As an example of a safety biomarker, hepatic aminotransferases may be used as safety biomarkers when evaluating potential liver toxicity. As an example of a response efficacy biomarker, HIV viral load may be used as a pharmacodynamic or response biomarker when evaluating response to antiretroviral treatment. As an example of a monitoring biomarker, hepatitis C virus ribonucleic acid, HCV RNA, may be used as a monitoring biomarker when assessing patients with chronic hepatitis C to evaluate disease status or burden. The convention in clinical research has been to measure the performance of novel therapies using clinical outcomes, such as mortality or disease progression. But accruing enough information to support clinical endpoints in a trial of a new drug may take many years. Some biomarkers can predict drug efficacy more quickly than conventional clinical endpoints. They have the potential to accelerate product development in certain disease areas, but the amount of data needed to understand these biomarkers as surrogate endpoints is much higher than the amount of data needed to support use of an enrichment biomarker. FDA has approved multiple drugs on the basis of biomarkers used as outcomes in clinical trials. CDER has published a table of biomarkers, categorized by therapeutic area, that have supported approvals of new molecular entities, NMEs, and new biological therapeutics in CDER between October 2007 and December 2015. FDA recognizes biomarker development as a high priority area for future research and collaboration among stakeholders and is taking action to better understand biomarkers used in drug development. Qualified biomarkers have the potential to provide valuable information that could reduce uncertainty in regulatory decisions at each stage of the drug development process. CDER's Biomarker Qualification Program strives to make biomarker data publicly available by establishing a biomarker's value for a particular context of use in drug development and regulatory review. Once qualified, a biomarker can be used in multiple drug development programs without a need for CDER to reconfirm the suitability of the biomarker for that specific qualified context of use. This program has the potential to advance public health by using biomarkers to streamline the drug development paradigm. We encourage drug sponsors to consult with the relevant review division as early as possible in drug development regarding the use of biomarkers in their planned clinical trials. Please consult CDER's website to learn more about biomarkers and to access additional information about biomarker qualification.
Hello, my name is Shayan Dasgupta, and I'm a biostatistician at the Vaccine and Infectious Disease Division at Fred Hutchinson Cancer Center. Today, I'm going to talk about evaluating the surrogacy of multiple vaccine-induced immune response biomarkers in HIV vaccine trials. Identifying biomarkers for use as surrogates for clinical endpoints in randomized trials is useful for reducing study period and cost, relieving participants of unnecessary discomfort, and understanding treatment effect mechanism. To understand a vaccine's effect in HIV prevention studies, considering models with a single biomarker may not suffice, and risk models with multiple biomarkers may need to be considered. Hence, our main interests of pursuit here are to optimize sampling scheme for biomarkers, to select markers with high surrogacy value from a list of many, to prevent overfitting, and to save cost of computation and maintenance of the data, and to build a parsimonious model increasing the predicted quality of biomarker combination. Award and notations. Let Z be the binary treatment indicator. Let S, S1 to Sj be a set of candidate surrogates. Let Y be the binary clinical endpoint. Baseline covariates W include demographics and laboratory measurements. SZ and YZ within parentheses denote the potential outcomes under Z for Z equals to 0 and 1. In our case, Z equals to 0 means placebo and 1 means vaccine. Frangakis and Rubin in 2002 defined the principal surrogate framework based on potential outcomes comparing the risk of disease in vaccinees versus that in placebo recipients at the stratification level S1 equals to small s1 and S0 equals to small s0. HIV vaccine trials, however, typically enroll subjects without previous infection, so S0 can be considered 0 for all subjects. Then, the task for principal surrogates simplifies to comparing the risk of disease within vaccinees versus that in the placebo recipients at the stratification level S1 equals to small s. So from now on, we will use S in place of S1 interchangeably whenever the context is clear. Also note that in a standard trial design, we do not know the potential immune response S1 for subjects in the placebo arm. Gilbert and Hudgens in 2008 defined certain identifiability criteria for the principal surrogate framework, and we will assume the same here. Number one is stable unit treatment value assumption. S1, S0, Y1, and Y0 for a given participant are independent of treatment assignments of others. Number two is the ignorable treatment assignment, which says that the Z, the treatment indicator, is independent of W, S1, S0, Y1, and Y0. We also assume that the risk of YZ given S and W follows a generalized linear model for Z equals to 0 and 1. Note that this third criteria is not required for identifiability of the principal surrogate framework. However, assuming a form of the risk model helps us in estimation purposes. Assumptions 1 and 2 guarantee that the conditional distribution of S1 to Sj given W in the vaccine recipients is same as that in the placebo recipients. This gives rise to the baseline immunogenicity predictor design given by Fallman in 2006, which says that utilize baseline predictors W correlated with S to build a predictive model for the missing values of S. Gilbert and Hudgens in 2008 considered a one-marker, one BIP model and showed that identifiability can be attained under the BIP design only after imposing an untestable constraint like beta 5 equals to 0 in our former risk model that we considered. Huang and Gilbert in 2011 extended the above setup by considering a combination of more than one marker in the model and proposed a semi-parametric approach to maximize the likelihood. In our work on this subject in 2018, we showed that under the multiple marker setup, the risk model becomes non-identifiable whenever number of markers exceed number of VIPs in the model. Thus, one way to solve the identifiability problem is by increasing the dimension of W. However, in a BIP design, precision of the principal surrogacy estimate depends on the strength of correlation between the VIPs and the potential biomarker. Thus, collecting multiple biomarkers may not be a practical approach as good VIPs are usually difficult to find. However, we can enhance the BIP design with a closeout placebo vaccination component giving rise to the VIP plus CPV design. In CPV, a portion of placebo subjects uninfected at the end of the trial receive vaccine at closeout and the response SC is measured. If condition 4 below holds as well, then we can substitute SC for S1 for the uninfected placebo group. And condition 4 says that S1 to Sj and S1c to Sjc are distributed with the same mean and error structures. In the BIP plus CPV design then, under conditions 1, 2, and 4, we can observe the conditional distribution of S given W in the vaccine recipients as well as in the uninfected placebo recipients. We also know phi naught W, which is risk of disease given W in the placebo recipients. With the knowledge of these three quantities, we can also estimate the missing conditional distribution, that of S given W in the infected placebo group. Thus, assuming phi naught W greater than 0, the BIP plus CPV design allows us to consistently identify all the conditional distributions in the model. With this additional condition 4, then, we can impute the missing values of S for the infected placebo group, and this lets us complete the dataset and use GLM to estimate the risk model parameters. In high-dimensional data, it is often desirable to have parsimonious or sparse representations of prediction models. So instead of maximizing the likelihood directly, we minimize this quantity instead, where L denotes the log likelihood given the data, P beta is a regularization function, and lambda is tuning parameter controlling the degree of regularization. Alternatively, we can also combine the mechanism of regularization with imputation in a bootstrap resampling approach. We call this procedure BIS or BISS, which is a general resampling approach that combines bootstrap imputation with stability selection. This was proposed by Long and Johnson in their paper in 2015, and this consists of the following steps. In step 1, we generate B bootstrap datasets DB based on the observed data DOBS. In step 2, we conduct imputation for each bootstrap dataset DB using an imputation method of choice. In step 3, we run randomized lasso on the imputed bootstrap data over capital lambda, a feasible set of regularization parameters, small lambda. And for the final model, we select parameters that are chosen greater proportion of times than the predefined threshold pi for any feasible small lambda and capital lambda. For the imputation step, Long and Johnson proposed Bayesian multiple imputation. We, however, propose to conduct the imputation step using the following algorithm. We call it the conditional imputation algorithm. This uses equation 2 to obtain the estimated probability distribution of S given W in the infected placebo group. That allows us to impute the missing values of S from this distribution and complete the dataset. Now we consider a simulation study with 4,000 subjects randomized 1:1 to vaccine and placebo with baseline covariate W, treatment C, and response Y. S1 to Sj are available for all individuals in the vaccine group and for all uninfected individuals in the placebo group. We consider a total of 25 markers, of which 2 markers have non-zero interactions with C. For each setting, 100 simulations are conducted, 800 bootstrapped imputed datasets for variable selection, and final risk model parameters averaged over 500 additional bootstrap imputed datasets. In our paper, we consider 5 simulation settings that differ in the following respects: number of main effects in the model that can vary between low, medium, and high, type of W with both continuous and discrete forms considered, and type of conditional marker distribution where we consider either multivariate Gaussian or multivariate exponential. In settings 1 to 3, Gaussian models for conditional distributions are chosen with continuous W, one each with low, medium, and high main effects. In setting 4, we choose Gaussian models for conditional distribution, discrete W with medium main effects. And in setting 5, we choose exponential models for conditional distribution with discrete W and medium main effects. In this presentation, we will show results from a few of these settings. The inference procedure consists of 3 steps. In step 1, we have to choose the form of the risk model. We use logic link in our simulations, but other links like probit can be chosen as well. In second step, we choose the form of the imputation model. 3 types of imputation models are considered here. The parametric and the non-parametric method uses the conditional imputation algorithm to estimate the missing conditional distribution, but either parametrically or non-parametrically. And thirdly, we also use mice. In the third step, we choose the variable selection model. We either use no selection, where no variable selection step is performed. We choose direct regularization, where multiple imputation is performed first, and then we use L1 regularization. And third is BIS, as described before. We also consider different types of performance measures. First is sensitivity, which is probability that a marker with truly non-zero interaction with treatment is selected. 2 is specificity, that probability that a marker with truly zero interaction with treatment is discarded. Then 3 is a weighted score of sensitivity and specificity varying with the weight W. The model prediction error, which is defined as for mu hat x, it is the square of the difference between the conditional expectation of y given x and mu hat x averaged over the covariate space. We also choose misclassification error, which is probability of being wrongly classified. Note that for BIS, value of pi which attains the best performance for each measure is also evaluated. Now we show results from some of our simulation settings. In setting 1, where we have Gaussian conditional distribution, continuous W, and low main effects, we see that all methods are performing really well when it comes to sensitivity. But in specificity, or in combinations of sensitivity and specificity, BIS with parametric imputation outperforms all others. It also achieves the best misclassification rate and best model error rate. In setting 3, where we have Gaussian conditional distributions, but with continuous W and high main effects, BIS with parametric imputation again achieves the best sensitivity and specificity score and combination scores as well. The misclassification rate and model error rates are similar for all methods. In setting 4, where we have Gaussian conditional distribution, discrete W, but medium n-effects, we use the non-parametric imputation method as well. It actually achieves the best feature selection performance, better than parametric imputation as well. The misclassification rate and model error rates are similar for parametric and non-parametric imputations. So summary of simulation results. All variable selection methods show high sensitivity in most settings. BIS outperforms direct selection in feature selection. BIS with parametric imputation outperforms BIS with imputation using mice in every setting. And the same trend is seen between parametric imputation and mice when used with direct selection. Now we come to a real data analysis where we apply the proposed methodology to data from RV144 study, an HIV vaccine trial that randomized 16,400 individuals, roughly half on placebo and vaccine. A follow-up immune correlate study assessed vaccine-induced immune responses at peak immunogenicity in a subset of vaccinees and identified 14 variables of particular interest. These variables were used to construct the list of potential biomarkers. Note that RV144 does not have a CPP component, so we use available data from RV144 to synthetically produce a CPP component. In Haynes et al., 2 markers were identified as correlates of risk for HIV. We generate the CPP component, assuming these biomarkers are true correlates of vaccine protection as well. Sex and baseline self-reported behavioral risk factors are used to create W, a BIP with 6 levels. Here are the results from the data analysis of RV144 trial. As can be seen, the non-parametric method is actually performing the best. It achieves really good sensitivity score as well as the highest specificity score as well, higher than the parametric imputation method. When it comes to cross-validated misclassification errors, it can be seen that all methods are roughly performing similarly. However, BIS with parametric method achieves slightly lower misclassification rates. Summary of real data analysis. So BIS performs quite well with regards to choosing both markers, and BIS with non-parametric imputation yields higher specificity followed by BIS with parametric imputation. For W between 0 and 1, performance of imputation methods decrease in the following order: parametric, then non-parametric, then mice, and cross-validated misclassification error are lowest for BIS with parametric imputation at pi equals to 0.9. Finally, discussion and concluding remarks. BIS procedure with parametric imputation is clearly the best performing method overall. Mice fails to correctly impute the missing potential marker values as it fails to utilize the randomization condition in equation 1. No optimal value for threshold parameter pi, so finding an optimal pi depends on the problem of interest. And finally, assumption A3 is not necessary for identifying disease risk model in a BIP or BIP plus CPP design. And these are the list of references. Thank you.
Great. Now, we will now have a 10-minute question and answer session. And I'm welcoming Ying Huang from the Fred Hutch and a colleague of Sayan Dasgupta who is joining us for the Q&A. So please raise your hand or alternately post your questions or comments in the chat box.
Raising my hand.
Yes, please go ahead.
Hello. Well, lovely presentation, Sayan, very clear, which got me lost after the first slide, basically. What is the one sentence home take message from your presentation?
So Ying, you're on mute, I believe.
Yeah, since Sharon is not available, I'm
sorry.
Okay, so yeah, so basically, I'll say this is really a project, a statistical project, trying to like finding a method to when we evaluate biomarkers. So basically, I think the general message is like a one single biomarker. If you think about like a surrogate marker for HIV vaccine is usually probably not adequate. So basically, we need two that can like trying to, because we have so many different biomarkers that measure in different labs, we really want a way that can combine biomarkers that can have us to get a better surrogate score. So basically, I think what Cheyenne has presented is really a project that's showing that like there is one statistical method that was developed for trying to combine different biomarkers that can come up with one good risk stratification score that can predict vaccines efficacy. And I want to say that there are different ways that can assess the biomarkers used, but what we use particularly here is like called principal stratification, so that based on potential outcome. And different statistics methods has different assumptions. So what we use here basically, because we use this potential outcome, potential biomarker, if you're receiving vaccine, it has some like missing data problem. So what this work does basically shows that there was one quite feasible method that can be used to select biomarkers. And also another thing is like, it's really evaluating biomarkers is a challenging problem. Like you have, if you use different methods, you're basically making different assumptions. So here, because we trying to not making this like unmeasured confounding assumption, we were using this potential outcome. So basically that posed like more challenging on study design. Basically what I think presented in the first half is saying like, if we have a standard randomized trial, it will be very hard to, there will be like identifiability problem. If we try to evaluate this type of principal surrogate marker, like combining multiple marker. So basically, there's a design called closeout placebo vaccination, like proposed originally by the informant, that says if uninfected placebo recipient at the end of trial, we can give them vaccination and measure their immune response, and use that to substitute their immune response at the like beginning of the trial. Then that one will help us to solve this identifiability problem. I believe this kind of like a trial component has been really now even used in, like, for example, in COVID vaccine trial. So that's kind of like, it's also considered like a beneficial to patients if the vaccine is really helpful. So yeah.
So may I have a quick follow-up question? So using this method, have we learned anything new about RV144?
I think what it's really for this, or it's basically it's a I would say like in this paper it's really kind of like trying to combine biomarker. Like in Cheyenne's work, he did some like showing that we can combine different biomarkers and try to will be a better prediction. I think it's kind of still it's an exploratory stage. It's not like validated really you have this combination, but really use like cross validation, we can show that like combining biomarker really getting like better surrogate score compared to like one single marker.
Yeah, great. And let's see, Deborah, you have your hand up. Give you an opportunity to ask a question.
Yeah, I do. Can you hear me okay?
Yes, we can hear you.
Yeah, you can hear me okay. Yeah, this is really not a very fair question, and this may not be the right part of the conference, of this workshop to ask this, but I'm going to ask it anyway, which is the principal surrogate approach that was presented. I believe this approach, well, this is really my question. Is it true that this approach is only useful in developing biomarkers in the context of having a placebo-controlled trial that has been conducted with a successful vaccine? And Linda Gale started us off with a statement, which I think is probably true, that, you know, placebo-controlled vaccine trials are probably not going to be able to be conducted. I mean, the field is yet to move this forward, but we do have long-acting products, one approved and another one under development, that would prevent HIV infections from happening in people, in the majority of people, I believe. I mean, I know this is all to be debated, but I guess the question to the statisticians is, if you are in a situation where you cannot get breakthrough infections on a placebo arm to compare to breakthrough infections on a vaccine arm, is there any path forward for developing surrogate endpoints or biomarkers that predict whether or not a person given a vaccine is actually protected?
I think like you might have, there's like no placebo arm for the.
Yeah. So like if essentially you could vaccinate people and observe whether or not they got infected, but you, I don't, who knows what the design would be, but like, let's suppose, for instance, you were doing a head-to-head comparison for a vaccine to cabotegravir. I mean, just make it up. So you weren't getting infections on one arm, even though they weren't receiving the vaccine because they were on cabotegravir. And on the other arm, I mean, I don't know how you would withhold with cabotegravir, but let's just imagine that you thought your vaccine was highly effective. And so you could observe people with breakthrough infections on the vaccine, but you don't have the infections happening on a placebo arm without a vaccine. You don't have breakthrough infections happening on the people who didn't get vaccines because they're protected by like a small molecule. I mean, so I just don't think, and I think this is probably what you meant, Linda Gale, is like, you know, I don't think we can go forward and have, you know, 50 infections observed in young women on a placebo arm, on a vaccine placebo arm, because we can prevent those infections and we know we can. So I just really was asking is like what's very difficult about this workshop in my view is like, do we have any way of developing biomarkers for vaccine prevention and without the presence of a group of people who are not receiving the vaccine and getting infected? Sorry, which is essentially a placebo
arm. Yeah. Interesting question. I have something like immediately.
I mean, it's a tough question, and it's like, it's not really related to statistical methodology. This is really sort of the design question. It's like,
Deborah, maybe, you know, the clinical trialist here could step in, you know, is there, so I can see the importance of having a non-vaccinated, you know, breakthrough infection, to be able to say, well, what did the vaccine do that protected people? So I'm wondering in future designs, if, you know, even though you have in your other arm very effective prevention technology, you do still get breakthrough infections because people may not take it, they may not persist, they may not show up, etc. So I think part and parcel will be collecting very careful histories from individuals across the arms to know what the use of product was, right? Because maybe it's in those individuals that you can then pick up the correlates to be able to do that sort of reverse engineering, as it were, which I can see that you're getting at. But I know we're going to be unpacking that more in our second part. So, you know, I think it's great that you introduced it. And I'll hand back to Carrie at this point to take us forward.
Great. No, Hanneke, you had your hand up if you had something brief, or we can.
I was just wondering whether if you have high diversity in your vaccine responses, whether you in your vaccine arm can compare high responders and low responders for relative risk of infection. Then you don't even need a placebo group, and you can probably prepare for that in your phase 1-2 studies where you see the diversity in immune responses.
Great. Yeah. So okay. So I think a lot of interesting questions. I think we'll get even more questions as we move on to the next section. So for the next section, we will now kind of move forward to our first group discussion on what could be used as biomarkers for efficacy in HIV in vaccine trials. And to set the stage, we will have some short perspectives from Larry Corey from the HVTN and Fred Hutch, Thomas Hanke from University of Oxford, and Hanneke Schumacher from Janssen Vaccine Prevention. So I'll turn it over to Larry.
All right. Well, I think I'm going to be a dissident on this one with respect to not sure that I agree with the basic tenet that you're going to have a counterfactual placebo group, or whether that's possible. I think the history of doing trials and has been to look at standard of care as a comparative arm. And I also would go back to the COVID vaccine program, including now, that there is a conservatism in licensing vaccines that are given to millions of people, and using a surrogate marker. I mean, we are 3.5 years into lots of effective COVID vaccines. We actually have a significant number of articles that have been written not only by our own group at the Hutch and CoVPM, but European groups coming up with a surrogate marker that's a reasonable surrogate marker that covers a significant number of the vaccines. And yet you have yet to see sort of the licensure of a primary vaccination, of a new vaccination just on surrogate markers. You're starting to see sort of approval of booster responses with a surrogate response. So there is this sense of, you know, the difference between a mechanistic surrogate marker, which you could say has been out of the monoclonal antibodies, in which you give a passive immunization, you give a single antibody, get a titer, you actually know that actually is a mechanistic marker, versus essentially a correlative protection. Neutralization is even at best the 50% predictor or 60% predictor of the mRNA vaccines, and somewhat less predictor of the adenovector vaccines. So if you take this body of knowledge and sort of say, gee, how are you going to look at a surrogate marker for an HIV vaccine, where frankly, we have not solved the immunology of that from a period of time. You know, we may have combination monoclonal antibodies, which would set a neutralizing antibody titer. Most vaccinologists would actually say to you that I'm not sure we have to actually reach the titer of a monoclonal antibody to get protection. That's actually true for symptomatic disease with COVID, where the monoclonal antibodies are higher in neutralizing titers than the mRNA, even when matched, and we got 95% protection, depending on what you want to call symptomatic disease. For HIV, we actually have to prevent acquisition, a higher bar, and probably one that requires more adaptive immune responses of some sort. And there's a feeling among most vaccinologists that adaptive immune response is multifactorial. So I honestly, I have my 3 minutes here to sort of say, hey, can you develop a biomarker that is going to predict efficacy for an HIV vaccine? And I would say at this point in time, other than really high titers of broadly neutralizing antibodies to multiple components, the answer would be no. I think it's not possible to do this priori efficacy.
Great, thanks, Laurie. I think that's a question, the chicken or the egg, in terms of the biomarkers and the clinical response, and we can have that discussion later on. So now I guess I'll turn it over to Tom for his perspectives.
Yeah, no, so I go very simple, basically saying that, of course, we haven't seen protection by vaccine, so it's hard to develop markers of protection if we don't have protection. And it's complicated for vaccines because we've got T cells and B cell inducing vaccines, we've got preventive vaccines, and we've got curing vaccine or therapeutic vaccines. And, these are complex, the protection will probably in the end be complex and combination of things. And for T cells, we can look at, magnitude, breadth, specificity, functionality, proliferative capacity, tissue localization, longevity of immune responses. And all these functions are important, and we need all of them to be optimal. One function of these, many, if it's not optimal, is going to cause that the vaccine doesn't work. So we have to induce all of these optimal. For broadly neutralizing antibody, certainly the level will have to be important. Specificity is important. Functionality of the FC, of course. FC is far from being constant. It can have 100 different forms, and each form will have different functionality, different activation complement, binding to different receptors, which their response is going to be combined, and again, longevity. So it's all quite complicated, and without having induced protection, or we just have no guidance for markers at this point, I would say. And I stop here.
Great, thank you, Tom, for your perspectives. And then now, Hanneke, your 3 minutes for your perspective.
Sure, thank you. So we have been working with partners on a prophylactic HIV vaccine, and we have used actually biomarkers in the program that supported our go, no-go decisions along the way. And the first ones were used to set criteria for human immunogenicity in the earlier trials to see whether we would proceed with the efficacy studies. And these were levels of cellular and humoral immunity that in combination correlated with protection in non-human primates against a repeat intrarectal shift challenge. And to just give a sort of picture of what we observed is that in that non-human primate study, that 11 of the non-human primates that had both high antibody titers and high level of ELISpot responses, 9 of these were still uninfected despite 6 challenges, whereas on the other hand, 25 non-human primates who had low ELISpot and or low antibodies. So it was really the combination or the lack of a response in one of the 2 compartments. Of these 25 non-human primates, only 3 remained uninfected. So we thought we really had some good idea of what we needed to establish in humans. Well, the Imbokodo study that we did overall did not show vaccine efficacy. So despite the fact that we selected for these immune parameters, it did not translate in vaccine efficacy in this population of women at high risk for HIV infection in Southern Africa. And this could either shed doubt on the choice of biomarkers, but it could also, of course, imply that the animal model that we used was not relevant, at least not for the at-risk population that we studied in Imbokodo. Now, having said that all, and while Imbokodo did not demonstrate an overall efficacy of the adeno plus protein-based vaccine, it did allow for a comparison of immune responses in women who had breakthrough infections during the study with the immune responses in women who remained uninfected during the study. And this was all studied in collaboration with HVTN, and Peter Gilbert did a lot of these analyses. But what is, first of all, very important to realize that you can learn a lot from efficacy studies, even those that have an overall negative outcome. But what I thought is very interesting, that the analysis, so comparing protectors from infected females, that it revealed that the breadth of V1, V2 responses was an inverse correlate of risk. So, well, that is then a standalone biomarker, but this observation does not stand on its own because it's sort of confirming the observations in 2 previous efficacy studies. So that non-neutralizing antibodies to V1, V2 are associated with protection from infection. And while I fully agree with Larry that it will be challenging to use a biomarker to really license an HIV vaccine, I think it is at least interesting to further explore the relevance of this biomarker and to see whether we can achieve higher levels of this biomarker in more people and see whether this then translates in significant overall vaccine efficacy. So I think really wrapping up, the biomarkers that we identified in non-human primates were not relevant to use for protection, to predict protection in humans. But I think the efficacy studies, although overall negative, did provide a lot of interesting information that really warrants further research, and that may in the end result in exploring the importance of this biomarker in overall efficacy.
Great, thank you, Hanneke. And then now we will open it up for discussion for the next about approximately half hour. So for those who have questions, for our panel, or please raise your hand. I guess I can lead off. I mean, Hanneke, like in terms of kind of the biomarkers that you did evaluate, for the clinical trials, you have to proactively collect the samples, but then you're not really sure what's going to be the right biomarker until you see kind of the clinical outcome. And so how do you think about that moving forward? It's kind of the whole chicken and egg program. We had this discussion last week at ASLD in terms of for hepatitis B surrogates of response and functional cure, and how do you select a biomarker if you don't know what the clinical outcome and how do you correlate that?
Yeah, of course, but that's of course also what Larry was pointing out, that you cannot predict what the relevant biomarker will be, so you cannot rely on it. I think what we now retrospectively did in the efficacy studies, because this was the Mokola study, but the same biomarker popped up in RV144 and in HVTN702, that collecting biosamples that will allow you to do these analysis retrospectively on what the potential correlate of risk is, or correlate of protection. That I think is the unique situation that we're in, that these three studies really took the effort to collect a lot of material to have deep analysis, and that's, I think, what I wanted to emphasize, that people always say it's a waste of money because right, it was not visiting broadly neutralizing antibodies, blah blah blah. But we learn a lot from these studies, and that these three studies have this biomarker in common, I think is very important. So prospectively, I agree with you. It will be very hard, and it's very costly because you need to sample a lot. Although you could probably take one time point at first to collect cells and serum. But and the other thing that I find really difficult to deal with is that for an HIV vaccine, as pointed out by Larry, we need to get protection against acquisition. I think that the timing is also, you cannot allow an incubation time. You cannot rely on recall responses. So to keep these antibodies up at sufficient protective levels in combination with T cells, I think that's really the challenging part.
Great. Mike, do you have your hand up?
Yeah, just really quickly, let me make a disparate comment in a way. It seems to me the discussion's too siloed, and maybe because the second half of this has more to do with monoclonal antibodies. The point is, the monoclonal antibodies give you a tool that allows you to do attributable risk of the benefit of non-antibody effects of vaccines. And so let me give an example of that. So what you see is Larry and Peter Gilbert and others have taken BNAbs and tried to show the concentration required for protection with an antibody. And that's now driving a lot of thinking in the HIV antibody vaccine field. But a stronger example is in the COVID field, where it's pretty easy for us to see how much antibodies being made by COVID vaccines and how much protection we get from monoclonal antibodies concomitantly, put the two things together into a witch's brew, and then come up with the appropriate surrogate that, in our opinion, probably would could lead to licensure. So what is my point of this? If you just take vaccines in isolation, you're missing this other tool that has become very popular in the COVID era. And I think that going forward, I would be surprised if these aren't married together very vociferously. I'll stop. Thanks.
But I would argue that taking antibodies in isolation, you miss a very important part of the protective mechanism as well.
Yeah, no, I'm not suggesting that. I totally agree with you. I'm only suggesting that when you give these antibodies, you can do attributable risk of the vaccine to antibody pretty clearly. You can see how much antibody it took. Then you can see the benefits of a vaccine. And Dean Philman's trying to write a paper about this right now. And then you can subtract that and say, well, the attributable risk to T cell and other host defenses is X, and this is Y. So I think antibodies are a tool to advance vaccine development. That's what I'm saying, and should be probably. So from what you just said, Hanneke, like I don't think you want to go back to macaques, because
No. But my point is that if you don't include the other components, you may end up with a too high bar for protective.
But I'm not suggesting. But I'm not suggesting that. I'm not suggesting you don't include. I'm suggesting that the animal studies may be less informative than rethinking this and using concomitantly kind of more modern tools, because the animals seem to endlessly disappoint us.
Yeah.
I'm not just in no way am I saying vaccines should not benefit from both limbs of the immune system.
Yeah.
I'm just trying to marry the two.
So, Mike, if I can just unpack what you're saying. So you're saying put monoclonal antibodies into both the control arm, if you like, and the active arm, and add the vaccine in the active arm, and then your standard of care arm is the monoclonal antibody.
Well, you could do that.
I don't think he's saying that.
No, that's not what I'm saying.
Okay, well, can you explain it?
Well, it's good that I'm not communicating well. Although I think that's an interesting thing. When you give a vaccine, as Hanneke pointed out, you get multiple benefits, including antibodies that are very clear in COVID. When you give a monoclonal, especially if you give a YTE mutation, the only thing you're getting is the antibody. So if you're doing those things concomitantly, if you're doing a vaccine for COVID, a Moderna vaccine, and a Evusheld monoclonal in similar hosts, you see exactly how much monoclonal you needed because it wanes, and then people start breaking through. And then with the vaccine, they're very interesting results because it's very clear vaccines do more than, they're better than the monoclonals in many ways, and they do more than the monoclonal can do. So it's really interesting, but I think what I'm saying going forward in clinical trials, these are tools that can be married together. That's kind of, so Larry, translate this for me. Did you understand?
Yeah, no, I think I got it now. Right.
I think translating it into a clinical trial is, however, difficult. I don't really see a clinical trial in which you give a subtherapeutic dose of a monoclonal plus a vaccine and sort of, how you and sort of how you look at that. I mean, I think Mike's point is, spot on when it comes to the issues of, what levels of monoclonal antibodies are required to, achieve X level of protection. We're still left with the conundrum that vaccines have a multifactorial adaptive and innate immune responses. And actually, we really haven't solved that. We haven't really solved it well for COVID, and we have not solved it at all for HIV. And I think we have a very illustrative issue with respect to what was learned from Imbicodo, and we'll have to wait and see a little bit what happens from Mosaico before we sort of totally throw out the non-human primate model as a predictive model of a biomarker. The problem with that model is that the challenge virus is not real. All right. And, that even if we believe the V1, V2, those challenge viruses, are not clade C challenge viruses. They're not the clade Cs that have drifted in the V1, V2 loop area. And maybe if we even look at the monoclonals for HIV, we find that clade C is susceptible to V2 loop antibodies a little bit more than clade B. Is less susceptible than clade B. So, there are some things that are difficult to conceive in an appropriate animal model with HIV because of its diversity. And as Hampus told us, we probably are affected with a swarm of viruses, not this illusion that we have one transmitted founder. So the breadth of what we need is still of importance. So, which does lead you back to the BNAB levels, but it's still the conundrum that we think vaccines don't, are multifactorial, and it's going to be very hard to do a biomarker when you really haven't solved the immunology. Post hoc, you can do it. Pre hoc, it's a guess. My opinion.
Sobering. So I guess, and it begs the question, so what is the way forward? I mean,
I do markedly disagree with you on this one, Linda, Gail, about that you have to have a counterfactual placebo. I don't see a road forward in any vaccine that you have to give millions of people without proof of a group that doesn't have the vaccine. Now, you can construct trials of standard of care, oral PrEP plus vaccine, oral PrEP alone, or maybe, it'll be long-acting PrEP, if something, or if that's the case, you go, you can sort of do a comparative trial. I think the issues are how you debate, what is standard of care. And I think your statement to some extent sort of says, gee, what we're doing in Mosaico of using people who want choice and elect consciously to not have an antiviral approach to prophylaxis shouldn't enter into clinical trial. And I would say that that's not ethical. That's exclusionary. And that's an issue that's been debated. TAG started that debate. We've had that debate for a long period of time. And I guess I would say to you, I think it's still operant. Tell me why it's not operant. Excluding people who don't want something, who under informed consent, seems to have issues as much as defining, a low bar on what's standard of care, or insisting what is standard of care. So I think there is tension in this field and always will be. And I think there's a diversity of experience that people want. And, and I'll just stop there and quit babbling.
Yep. Richard, you have your hand up.
Yes, thanks. Oh, might as well see me. Nice to see you all. Two very simple-minded comments, actually. I mean, firstly, I agree with the point just made by Larry about, the importance of giving potential trial participants choice. And, I think one can make a sound ethical argument that if trial participants have been given the choice of accepting a preventive measure that's been proven and they refuse that choice knowing what they're doing, then that probably is ethical. I think myself that cabotegravir by injection has made this a hell of a lot more difficult. I mean, it's wonderful that we have these cabotegravir results. We really needed it, right? Given the challenges of delivering oral PrEP, which I think we're all familiar with, especially in high prevalence settings as well. Now we have this injected product, for which the challenges of adherence and so on are much, much less. And especially in countries where, at least for family planning, injections every 2 or 3 months is highly acceptable. And in fact, it's like the contraceptive of choice for many women, perhaps less so for men, of course. But the problem is now we have this wonderfully effective product that it looks as if efficacy is in the high 90s. So it's almost completely protective. And it's something that you can monitor giving it so well in a trial setting where you're following people up, very carefully. You'd be seeing them routinely in the clinic, periodically. And so it will be very easy to provide that intervention. And my suspicion is that it's going to become increasingly difficult to find participants who, given the information about that protective measure will actually decide not to accept it, particularly if they're living, as they would have to be, in a population where there is a significant risk of HIV. So I think just in practice, that's going to be a really big challenge. And to be honest, I think this is a really major challenge now for future HIV prevention trials. My second comment is even more simple-minded, sorry, and forgive my lack of knowledge of this field of vaccinology, but we've heard that at the moment, we don't have proven surrogates of protection, basically to a large extent because we haven't had a successful product that's been proven through clinical trials. But suppose even that we did, suppose that we did have a vaccine trial that had shown clear evidence of protection, and based on the data from that trial, we've developed a surrogate measure or a combination of them, as we've heard today, perhaps a combination of 2 or 3 measures that are highly predictive of protection. But then going forward to the next products, which may well work through different immune mechanisms, can we still be sure that any surrogates that we use would be appropriate for a different vaccine product? So that's another thing that worries me. How specific to particular vaccine products would any surrogates be? Thanks. Those were just my 2 very simple-minded comments. Thanks.
Yeah,
Well,
no,
yeah.
Go ahead.
It reminds me of this study in, I think, Nature Medicine, where after stopping antiretroviral treatment, there were two individuals, and one was controlling the rebound by T cells, and the other was controlling the rebound by broadly neutralizing antibodies. So yeah, there are going to be different individuals who will have different pathways to protection.
Yeah, no, I think that's a major challenge because, with the different technologies that are being used in terms of, kind of the vaccine strategies from mRNA to adeno to, we have a CMV HIV program here at VIR. I mean, they all elicit different types of immune responses, and trying to correlate that across different, populations and trials. And it will be, I don't think you can apply one to the other. Maybe open it up to some of our industry colleagues who are on this call from either, I think ViiV and Gilead and Merck I've seen, familiar names. So if you guys have any comments on this section as well.
Hi, so this is Elizabeth Wunderlich, and I'm calling in from ViiV Healthcare. And I think overall, the entire discussion is spot on when it comes to right now, we don't have a biomarker for efficacy, and it's really hard to find a biomarker when we don't have protection. So if we don't have effective vaccines, it's difficult to have a biomarker for efficacy. And I would even, look back at COVID as an example as well, because, even though we're getting T-cell responses and BNAbs, people are still being infected. Yes, it's absolutely decreasing severe disease, hospitalizations, and deaths, but it is also not necessarily considered protective from infection. And so with HIV being different, because you form a reservoir and it ends up being a long-term infection, it's a little bit different beast, but I would even argue that we don't necessarily have a correlate of protection yet for COVID, because we are still being infected despite T-cell responses and antibody responses as well. And so this debate over what correlates of protection would be the best, I completely agree with Tom and Larry. It's really hard to try to define them, but that's the business that we're in, is trying to find that treatment that will provide efficacy, that will provide that biomarker, so that we do have some kind of gold standard going forward. So we definitely continue to look into these biomarkers for relations to efficacy. And coming from the company with the long-acting cabotegravir, we're extremely happy and excited about this product, and definitely see how it's throwing some wrenches in some clinical trial designs, but it's such an amazing product for so many people around the world that I think we're all willing to have to deal with those wrenches that are being thrown in there. So but overall, I think everybody's, you know, an ideal situation, like Hanneke was talking about, with, having a range of efficacy in the non-human primates, that would be a really great thing to be able to have happen in human clinical trials, but it's still a little bit of a hope to be able to do so, to have these sort of outcomes in human clinical trials. So, I think with RV144, we are looking at those, the higher end concentration of individuals having better protection, and things like IC80s and IC90s coming out as being important for sensitivity to BNABs. And so hopefully we'll be able to continue down that process of taking, the tails of normal distributions, taking the tails for these clinical trials, that as we keep going, maybe, if we, the evolution of biomarker plans and efficacy is that we'll keep taking those, the 5% top end cases and have those help us continue to drive the biomarker discussion. So I think I see James Rooney has a question.
Jim, I think go ahead, Jim. You're muted.
Yeah, no, still trying to work out the technology, Linda Gale. Sorry about that. I'm on my phone. So I can't get my hand down. That's what I was trying to do. But anyway, I just want to mention that certainly if you could identify a surrogate of protection, that would be ideal. Obviously, for other vaccines like influenza, et cetera, that's the standard method for approving new ones without having to go through a clinical trial. And certainly encourage continued investigation in the field. I just wanted to mention as well, if indeed it ends up being too challenging, and for example, as has been noted already, there's different immune mechanisms that might be responsible for protection, either in different individuals or different with different vaccines, different populations. An alternative method to evaluate the efficacy of a prevention intervention would be to look for an alternative estimate of infection. And as many of you know, the current prevention studies that are ongoing now, looking at long-acting agents, particularly Gilead's study and also Merck's original study with islatravir, we're using, it looked very carefully at this over a period of time, and populations we were examining, it turns out that there were alternative methods of assessing it, either by looking at infections in certain populations, rectal gonorrhea and MSM, much more difficult in high-risk women to look for an infectious correlate of incidence. But recency assays have emerged as a potential strategy in that space, and certainly have gone through at least initial regulatory review with tentative agreement from regulators in sub-Saharan Africa and also in the developed world that it's an approach at least worth evaluating. It hasn't really been done in the field yet, and there are potential significant limitations to its success, but nonetheless, it is a strategy that's being explored for other prevention interventions, and eventually I guess could be one that could be used in the vaccine space as well. So anyway, I just wanted to mention that as a parallel effort ongoing in a separate, in similar endeavor, and something that we should all keep an eye on. I think we all share the need to come up with a new strategy for doing prevention studies, and certainly we're all interested in trying to identify what that might be. So I'll just leave it at that. Thanks.
Okay. Thanks, Jim.
Now, thank you.
Yeah,
Yeah.
another sorry, just quick. Another quick note is something that Larry mentioned was a mechanistic marker as opposed to correlative protection. And that was something that, you know, I'm really taking home with me to ViiV is, you know, correlative protection has been elusive for a lot of respiratory viruses in particular. So I do have a history looking at influenza and COVID and RSV as well. And so, you know, overall, we don't necessarily have the greatest handle on a correlative protection for respiratory or mucosally acquired viruses. And so I think, you know, trying to find the strongest mechanistic markers is a really good way to try to move into hopefully finding one of those mechanistic markers that does end up being a correlative protection. But it's definitely going to be and has always been and will be until we have one. It's a process of defining those and finding which ones work best until we find the next one that works best. So yeah.
Great. Yeah, no, I think it's clear that there is not, you know, clear 1, 2, 3 biomarkers that can help predict kind of response to vaccines. But I think from an industry perspective, I mean, there's 2 different views on how to approach this. With biomarkers, it can be used to kind of evaluate your platform to see if you're elucidating the type of responses that you're looking for before you take it into the clinic. Because obviously these large clinical trials are quite expensive to do. And so if we can identify biomarkers to help select which, you know, vaccine platform to move forward with, I think that's definitely helpful. And then, you know, if we get something to the clinic, you know, I think the higher hurdles from a regulatory perspective is trying to use these biomarkers from a regulatory perspective that as using as a surrogate for protection. I think we would still need to see a clear correlation between kind of the clinical prevention of HIV infection and, you know, being highly correlated with the biomarker itself. So.
Yeah, I completely agree that, yeah, you know, based on efficacy that we're seeing right now and how you move platforms forward. And then from there, how it relates to protection and, yeah, disease control, theoretically, if we get into therapeutic vaccines. So, yeah, I very much value everything that's been discussed so far, and in particular the biomarker validation platform from the FDA. I think, you know, I think that is kind of the community thought that can help us move towards more correlates of protection and more effective outputs. So I think, you know, I'll look into how ViiV is able to help and contribute there.
Thank you. So I don't see any other hands raised or any other questions in chat. So I think, you know, now's a good time for a 10-minute break. So thank you to all our speakers for this first panel discussion. I appreciate your participation and everybody else's participation. So we will take a 10-minute break. Let's come back at 35 after the hour. Since I don't know which time zone everybody is in, but I think we can go 35 after the hour and we'll restart at that point.
Let me just very quickly jump in here, following what Carrie said. Indeed, please do come back for part 2, in which we will discuss examples of biomarkers recently identified and how these can be used in trials. We will also hear from a regulator's perspective, so this follows up from what Carrie also said earlier. So looking forward to seeing you in part 2 in 10 minutes. Thank you, everyone.
All right, Carrie, thank you for the moderation. Same, Linda Gale. Thanks.
Thank you, Larry. Thank you.
Okay, keep on, Carrie.
So welcome back, everyone. We will now start the second part of this workshop on using biomarkers as surrogate endpoints in HIV prevention efficacy studies, challenges and opportunities. I will hand over to Linda Gale to introduce our speakers.
Thanks, Julie, and welcome back, everyone. Thank you for continuing, I think, an interesting discussion as we continue to engage around the challenges, but also investigate some of the successes we've seen recently. And the way we're going to proceed in the next section is with two expert interventions. So we want to thank the speakers for participating. We'll then have a Q&A. And so again, I ask you to, list your questions either via the chat or be ready to put up your hand and verbalize. We'll then move into the kind of moderated discussion with three varying diverse opinions with short interventions. And then we hope a very, interactive discussion to end us off. So to start with, it is my great pleasure to introduce somebody who I think is, blazing a trail in this field and is a co-author together with Peter Gilbert on the work that was recently published in Nature Medicine. So Yunda Wang, friend and colleague from the HVTN Sharp group, and she co-authored this paper, PT80, a neutralization titer biomarker for antibody-mediated prevention on HIV acquisition. Absolute delight to have you, Yunda. Over to you.
Thank you, Linda Gale, and thank you for the opportunity to share what we have learned in the AMP trial using PT80 as considered as a biomarker for antibody-mediated prevention against HIV acquisition. Let me see. Can you see my slides okay?
Yes, it's not in the presentation mode, for me at least.
Okay.
Yeah, we're seeing the speaker screen with the.
Okay, let me 1 second. Somehow my computer is slow this morning. Okay. Can you see the folds right now?
There. Perfect. Yes, wonderful. Carry on.
Okay, thank you. Yes, so I will start the presentation with a brief overview of the antibody-mediated prevention AMP trials, summarizing the prevention efficacy of VRC01 as the intervention in the AMP trials, and follow with presenting results on studying biomarker of prevention efficacy and with an example of using the biomarker for the design of future trial. As you all know, the AMP study examines the prevention efficacy of VRC01 that targets the CD4 binding site on the HIV trimer, and the primary results have been published in March 2021. Just briefly, so the AMP trial consists of two parallel trials with 2,700 conducted in the Americas and Europe, and 1,900 in the Sub-Saharan Africa, with a total of 4,600 participants who received 10 infusions of VRC01 in either low or high dose or at the placebo control, with infusions every 8 weeks, with a total study duration of 22 months. Although there was no overall prevention efficacy observed in either trial, other study population, however, in the pre-specified analysis, we have observed estimated 75% efficacy of VRC01 against viruses that are neutralization sensitive to VRCO1, where sensitivity of the virus is defined as IC80 less than 1 microgram per ml, meaning that the concentration of VRCO1 is less than 1 microgram per ml to reach 80% neutralization of an acquired virus from participants, isolated from participants who acquired HIV during the study. And we have also, in addition, we have also seen from the M trial, from both studies that, that the prevention efficacy decreases smoothly with resistance level, i.e. the IC80 value of the concentration against the acquired concentration VRCO1 against the acquired virus. However, IC80 is on its own is a property, is a neutralization property of the BNAb, the clinical lot of the BNAb against a given virus. It has nothing to do with how much of the drug is in a BNAb recipient. So IC80, although we have seen this curve of prevention efficacy against IC80, this perfect relationship, IC80 itself is not a sufficient correlate. As you can see as an example that it has nothing to do with the concentration of the BNAb in a participant. So you see basically no prevention efficacy against even a sensitive virus with IC80 less than 1 microgram per ml when there was no VRCO1 detected in participant sera, for example, at the right tail through the 32 weeks post the last infusion in the M trial. So we proposed in the paper that be published in Nature Medicine using the predicted 80% inhibitory dilution titer we call PT80, the predicted ID80 titer, as a marker to predict prevention efficacy, because it is PD80 has a proper neutralization property of both the serum sample of a BNAb recipient at a given time. So the person has a given concentration level of a BNAb that received. At a given time, the participant sera has the titer against a given HIV virus. So mathematically, we can calculate the serum PD80 titer as long as we know, or we can predict the serum neutralization serum BNAb concentration in the participant serum in a serum sample of a BNAb recipient. I use that divided by the IC80, which is what we'll be talking about. And so we can predict the predicted ID80 titer of the serum sample. So the PD80 itself can be a sufficient correlate because it is has a property of both the host BNAb concentration as well as the virus, this neutralization sensitivity of the virus by the BNAb. So as you know, we not necessarily can experimentally measure the serum concentration at any given time, especially at the time when a person were exposed to a given to a virus. So we have to predict the serum concentration of a BNAb at any given time. And so we have published several papers that have shown that predicted PD80 titer is a good predictor of experimentally measured serum ID80 titer. So you can see in this graph at the lower bottom here, where on the y-axis shows the observed and the x-axis shows the predicted ID80 titer using this formula, where the concentrations are being measured experimentally, and the observed that ID80 titer is also being measured experimentally. So you can see the predicted ID80 titer can very well agree with the experimentally measured ID80 titer. So with that in mind, so we presented the results using the PD80 as a biomarker for the antibody-mediated prevention of HIV acquisition And in the paper, we described that the prevention efficacy goes smoothly up with the higher with the PD80 value against the acquired virus. So basically, this is similar to what I just showed you, where the prevention efficacy against IC80, except that we also scaled IC80 on the x-axis by using the concentration and the median mid-infusion visit divided by the IC80. So this is a population level approach in that it does not necessarily count individual ID80 titer as the challenge. But in this relationship, relationship between prevention efficacy and PD80, we can see suggests that at ID80 titer, PD80 titer around 80, we can see around 75% prevention efficacy around 90%. And it was also confident to see that this relationship between prevention efficacy and PD80 titer against acquired virus is also seen consistently in non-human primate studies. And we have done our group had done a meta-analysis of 256 animals who received more than a dozen of BNABs, single BNABs in these studies. We have captured the relationship between the prevention efficacy observed in those non-human primate studies versus the predicted ID80 titer against the challenge virus. And you can see the black curve just repeats what you just saw for the from the AMP trial, and the color curves just represent different subsets of the non-human primate challenge studies have captured. And we all know that although it was very amazing that in AMP trial, we were able to see just using a single BNAB that 75% prevention efficacy was observed for single BNAB against neutralization sensitive viruses. But we also know research has shown that probably multiple BNABs will be required, targeting the HIV trimer will be needed for achieving even higher prevention efficacy at a relative lower feasible dose amount of the BNAB. And now next, I'm just going to show you an example of such combination BNAB consisting of 3 BNABs targeting respectively the CD4 binding site, the V1, V2 apex, and then V3 glycan site, consisting of VRC07, 5-3LS, PGDM 1400, and PGT 121. So with this combination, we show that the shape that with the curve, this is the black curve that you've seen based on AMP study. And we have made additional assumption that the green curve based on the non-human primate study that studied BNABs that targeting those 3 epitopes on HIV trimer, that we have made conservative assumptions that it would require even 50% higher titer in order to achieve the same level of prevention efficacy as we observed in those non-human primate studies. And you can see this curve shifts a little to the right, even compared to the curve we observed in AMP study. And so here, just briefly summarize how we did this exercise. So basically, as you see from the AMP trial, we observed this curve relationship between prevention efficacy and PD80. And then for a combination BNAB, we can also predict the predicted ID80 titer over time for a given dose of each of the BNAB in the combination. And then putting these 2 pieces together, as well as incorporating the hypothesis distribution of HIV exposures, as well as the transmission probability estimated from historical data in the study population of interest. Putting these pieces of information together, then we can look at the prevention efficacy of the combination BNAB against each specific circulating virus. Here, I'm just showing 2 examples of viruses. The green shows sensitive and the red shows resistant virus. But in reality, we can basically have a single curve for each of the virus that a given participant will potentially be exposed to. And then with the information on the distribution of the viruses in terms of the IC80 against the BNAbs that we are studying, then putting these all together, when we look at the viruses, especially as a representative viruses from the study population, in examples that I will show, we'll use the viruses that we isolated from placebo recipients who acquired HIV in the AMP trials. And with these pieces of information together, then we can predict the prevention efficacy against across all circulating viruses in the study population of interest and at any given time. So with that framework, that as an illustration, here I'm showing you the predicted prevention efficacy for the triple combination I just mentioned. We here for illustration, we looked at two different dose levels, the higher dose level, 40 mg per kilo dose level for each of the BNAb in the combination, and the lower dose of 20. And for comparison, we also put in context the VRC01 prevention efficacy over time had it been given at a higher dose than what it was observed, what was used as higher dose in the actual AMP trial of 30. And you can see that when we apply the framework that I just described to the panel of viruses that are isolated from the sub-Saharan African trial participants who acquired placebo recipients who acquired HIV in the trial. And we can see this is the predicted prevention efficacy over time. And we did the same exercise against the primary clade B viruses that are isolated from our 704 AMP trial placebo participants. And in summary, we're seeing an average prevention efficacy for within this 16 weeks, and we've done the exercise for 24 weeks as well, that over 90% prevention efficacy could be predicted is expected for these two triple BNAbs at the low and higher dose levels. So I think it's wonderful that we have this biomarker PD80 that we can potentially use as a tool to rank and select BNAb regimens for future efficacy testing. At the same time, there are some open questions. So the PD80 correlates results that I just presented in AMP study may or may not transport to BNAb regimens targeting non-CD4 binding site epitopes in humans. VRC01, we've seen very encouraging results of 75% efficacy against sensitive viruses, against VRC01 sensitive viruses for a single BNAb targeting CD4 binding site, but we don't know whether BNAbs targeting other epitopes, B3 epitopes on the trimer, whether or not it will require lower or higher PD80 titer than the CD4 binding site targeting BNAbs that we observe in AMP trial. We also don't know, so AMP trial also only use a single BNAb regimen. We don't know for BNAb combinations, for example, the one that we just discussed, whether these combinations, which each of the BNAb component in the combination targeting different epitopes, whether or not it will require lower or higher PD80 than a single BNAb. In the exercise I showed, in the combination titer, would it require at the same level that as you would for a single BNAb, assuming that these BNAbs in the combination would work complementarily in terms of neutralization function. But whether or not this will be true will need to be validated and tested in a clinical trial setting. There are also other parameters other than PD80 that may be predictive of prevention efficacy, could be considered for the prediction of prevention efficacy in humans. For example, for some viruses, not necessarily 100% neutralization can be obtained for a given BNAb. And so how much of the extent of incomplete neutralization could contribute to the prediction of prevention efficacy of a BNAb regimen that will need to be studied as well as the neutralization curve slope in terms of how fast the neutralization the concentration of the clinical lot of VNAP needs to increase from neutralizing 50% of viruses versus 80%, in terms of how steep that neutralization curve is and how that predict. That's also an open question. So with that, I'd like to especially thank so many people, and especially our trial participants, contributed to the AMP study and our AMP study protocol team, our chairs, co-chairs, Larry and Mike, and also everyone on the team, and especially want to thank my colleagues, my statistical colleagues, Peter and Lady for their comments on the slides. That concludes my presentation.
Thank you. Thanks, Yonda. Thank you very much. I think we appreciate you taking us through that terrific paper and the progress that was made in understanding the contribution of the AMP study. So thank you for that. Again, I'm going to ask folk to hold on to their questions because we're going to take the next intervention, and then Yonda, if you'll come back to answer any questions in the Q&A. So let me introduce our next speaker, and it's my pleasure to introduce you to Marco Cavalieri, who's the head of health threats and vaccine strategy at the EMA. So this enables us to hear a regulator's perspective on the use of biomarkers as surrogate endpoints. So Marco, over to you.
Thank you very much, and hello to all of you. I will now share my screen. Hope you can see that.
Yes, and
Okay.
if you just put that, perfect. Thank you.
Very good. So I will give you a bit an overview of regulatory agencies looking to the use of biomarkers as surrogate endpoints for regulating vaccines. And I will start with the toughest scenario, but also the most important one, which is the use of biomarkers as surrogate endpoints for an approval of a brand new vaccine. Normally, this is possible if an immune course of protection is available, and I will say more about that, what we mean with that. But it can be also possible if you got an immune marker that is suitable to infer protection and will be applicable, and field efficacy trials are not feasible. So there are also options in case we don't have a fully validated immune course of protection, still to use a biomarker as a surrogate endpoint to support approval. And then there are scenarios in which none of the above is possible, and maybe not even field efficacy trials, and then we need to be really creative, and that's where we need to discuss with developers what could be the options there. But I will start indeed of the scenario, the most traditional one, which is the approach when you got an immune course of protection. First of all, at the EMA, what we mean with an immune course of protection is simply an immune parameter that has been demonstrated to correlate with protection at a defined value. So we have a threshold, and we have data that really support the fact that that threshold could be used to distinguish those that are protected and those that are not. And established courses of protection do exist, as you know, for example, for tetanus, diphtheria, polio, Haemophilus B, and hepatitis B, for example. Normally, an immune course of protection is derived from phase 3 studies, and as you discussed also before, with the first-in-class vaccine that runs a study versus placebo, and you really look into, once you achieve efficacy in your study, you look into immune markers in those that participated in the clinical trial to see if there was any difference in the amount of any of this specific biomarker that could really indicate what could be the immune course of protection. However, there are circumstances in which effectiveness studies or natural infection seroepidemiological studies could provide evidence on course of protection. And I will give you an example. Right now, we are discussing the possibility of approving chikungunya vaccine. No vaccine for chikungunya has been approved so far. And we already seen a number of these seroepidemiological studies that really tells us that people already exposed to the virus, that they are protected. And therefore, we will be keen, based on the solidity of this observational study, to really use such kind of marker in a scenario like that one, so like for chikungunya. But of course, not forgetting that chikungunya from an immunological perspective is not as a complicated case as many others such as HIV. And in some cases, we do not exclude that also human challenge studies, which are becoming more popular, and we will see hopefully, an increase in the use of these models, at least for certain pathogens. Could also help us in identifying what could be a course of protection. But also animal models of infection are important and always provide valuable input with respect to what could be a course of protection. What is important anyway to remember is that courses of protection, even once they are established and maybe in a very convincing way, may not be applicable beyond the vaccine and the population in which it was identified. So the mechanism of protection might not be the same for vaccines that are based on different platform technology and or target different type of antigens or the same pathogens, and possibly leading to a different kind of immune response. So we have to be very careful on the fact that once a course of protection is established, does not mean it can be used across the board for whatever type of vaccine is under consideration. Also, the route of administration can play a role. So, for example, oral versus intramuscular could have an impact because it changes the way the antigen is presented and the immune response that will be generated. And also there could be differences across population, even if this is quite rare, but we cannot exclude that in certain population there might be some differences with respect to what are the immune markers that are really impacting on protection. And the last but not least, also different syndromes due to the same pathogen may require different immune course of protection. For example, for pneumococcus, prevention from invasive disease is very different than prevention from otitis media or pneumonia. So it's important to consider that for different type of syndrome in different body location, for example, or with different type of penetration deep into the body might require special consideration, and you cannot use the same course of protection across all these different syndromes. So with respect to the pivotal study design that should be considered here for in case you've got a course of protection established and available. Normally what we look at is the pre-specified post-primary response, which usually is defined as post-vaccination seroprotection rate, so the percentage of subjects with an immune response that is above the threshold defined in the immune course of protection. Randomized controlled trial study to look into testing non-inferiority versus a licensed vaccine in terms of seroprotection rates is what normally we see conducted, also because you have the opportunity to compare the safety. But also when you have a real and established course of protection, you could even go into a single arm trial without the need of having a comparator vaccine included in your study, because what matters is the level of seroprotection and the rate of seroprotection you can achieve with your new vaccine. Very important as a regulator to stress that full validation of the assays is needed, including performance characteristic. This is crucial and has to be provided. And also for post-primary booster vaccination. So in this case, when you got an amnestic response, then the geometric mean concentration and titers or geometric mean ratio might be appropriate primary endpoints. And these very much depend on a case-by-case basis. But there are differences in this type of setting compared to what would happen with a primary series. And again, this is just an example that, this comes from one vaccine that we approved not long ago is a typical hexavalent vaccine for infancy, in which you see that the majority of the antigens included in this vaccine, like diphtheria, tetanus, polio 1, 2, and 3, Hib and hepatitis B, for which you see indicating what is the established course of protection. We're meeting the criteria of very high level of seroprotection, as you see here on the rates according to different schedules that have been used. And this is an example of how we would use in terms of approving new vaccine an established immune course of protection. Then there are other situations in which we don't have an established immune course of protection, as mentioned before, but there is an immune marker that is suitable to infer protection. And in this case, might be that we don't have indeed an immune course of protection, but we have data that points towards a threshold value that appears to correlate with protection and could be used, therefore, as benchmarking the level of immunity achieved by a new vaccine compared to an already approved one. An example here is the level of immunoglobulin G elicited by conjugated pneumococcal vaccine for specific serotypes. And this type of approach has been used also to license second generation pneumococcal vaccine. If there is no immune course of protection or threshold for benchmarking immunity available, it could still be possible anyway to use an immune marker that best represents response to a vaccine that showed efficacy. And an example of this kind is what we're doing with the cellular pertussis vaccine, for which there is no threshold. We don't have even a kind of rough estimate of a value that could be used for benchmarking, but we know that immune response to certain specific antigen for pertussis can be used in order to bridge immunogenicity from one vaccine to the other, or at least immunogenicity from an approved vaccine that demonstrated efficacy to a new one. And in this case, in a situation like this, the comparison of the immune response between the candidate vaccine and a licensed one for which efficacy or effectiveness has been shown would be expected, as just mentioned. And again, if there is no threshold available, the primary endpoint is usually the seroconversion rate, which is meant as a predefined increment in antibodies from pre to post vaccination, but also in some cases, the geometric mean titers of the antibodies. Generally, we as regulator, we would like to see functional antibodies over binding antibodies. And also here, we would need to see full validation of the assay that is used in order to determine the neutralization. T-cell responses, we know that they are extremely important and always have a role and an impact in terms of the level of protection you can exert, at least for many pathogens, maybe not all of them, but for many for sure. But the problem is very difficult to use them as part of the inferential testing for immunobridging. And so far as regulators, we've never seen a single case in which T-cell responses could be used for regulatory purpose as a potential biomarker that can be used for inferring protection. But of course, it's always good to try. We hope that there will be new technologies and we can do better here in determining exactly what is the impact of T-cell responses on the level of protection. And I will give you an example that comes directly from COVID. And this is a paper from Peter Gilbert that I know is attending this meeting. You see here that essentially for COVID-19 vaccine, there is a clear correlation on neutralizing antibodies with protection from symptomatic disease, because again, it's very important to distinguish protection from infection versus symptomatic disease or severe disease. They are not the same, but from symptomatic disease, which was the endpoint of the pivotal clinical trials, like here for the Moderna messenger RNA vaccine that was used for this analysis, there is a clear correlation between the amount of neutralizing antibodies elicited by this vaccine in a primary series versus the level of protection from symptomatic disease. But what you see on the right is that when we look at cases versus non-cases, you see that it's not possible to identify a clear-cut threshold that would really differentiate those that are protecting than those that are not. So in other words, there is a correlation with neutralizing antibody, but no threshold available at the moment, and maybe will never be the case. And again, looking at SARS-CoV-2 and vaccine for COVID, as also here, the T-cell responses, we know they are very important, that they provide a lot of advantages also in terms of protection in the long term and against variants of concern, as this paper was nicely illustrating. But the problem is that, again, it's very difficult really to use assays that can be fully validated and very well interpreted in terms of what exactly T-cell response would mean and how much they would impact on correlation to protection. And lastly, but not really a minor point, that is very difficult to disentangle then the contribution of T-cell responses from the humoral response, which of course is also very important for protection from severe disease, as monoclonal antibodies has repeatedly shown. So here we got a challenge really in not being able even today to even consider using T-cell responses as part of our currents of protection for regulatory purposes. And this is a case of how we use the immunobridging, again, in the context of COVID-19, based on the principle that indeed there is a correlation between neutralizing antibodies and protection. And therefore, we would assume that if a new vaccine is able at the population level to reach the same level of neutralization as vaccine that demonstrated efficacy and well licensed, then we will be in the position to authorize this vaccine. And in this case, because the comparator could not be the most potent vaccine that we have for COVID, but was an adenoviral vector vaccine, which has somehow lower level of neutralizing antibodies and also efficacy, here we demanded to show superiority to that vaccine that anyway was approved based on positive result in terms of protection. And we use this path for approving this vaccine as a new vaccine without the need of conducting clinical efficacy study. And very recently, we also approved a vaccine from Sanofi Pasteur, which is based on the beta variant. This has been approved as a booster because it really demonstrates to be better than community, so the Pfizer-BioNTech monovalent original strain vaccine in terms of neutralization of Omicron subvariants. So again, we will be using systematically immunobridging for new vaccine for COVID-19. But it's also important to think that biomarkers, I described to you what could be, the use for the sake of licensure, which is probably the highest bar scenario. But biomarkers and immune markers are extremely important throughout the life cycle and the development of new vaccine. And the level of validation and establishment may vary depending on what is the purpose. And for example, biomarkers are extremely important for selecting dose for new vaccine, understanding whether you need an adjuvant or what could be the impact of an adjuvant in early clinical development can be extremely important to understand what is the impact with respect to co-formulation with other antigen or co-administration with other vaccine. Important to understand what is the impact on the route of administration, the response you get with the booster shot, and lot-to-lot consistencies or any change that may occur during the life cycle in terms of the vaccine composition and how this is done. So we should not forget that immune markers and biomarkers for vaccines are really vital, even if they are not fully established depending on the purpose, but they're really vital in order to move on with the life cycle management of the vaccine. And another example, again, coming from COVID-19 is the use of the immunobridging, to extend the approval across different age ranges, because maybe the initial clinical trials that demonstrated the clinical efficacy in the field were done with a specific population and a specific age range, and you may want to extrapolate here like what to do with the pediatric population, and you can use the immunobridging approach here in order to allow the approval of vaccines across different ages that were not studied in the clinical efficacy studies. So this is my last slide. And just to recap, if an immune course of protection is available, clinical immunogenicity data will be sufficient for approving a new vaccine. In case there is not a course of protection established and field efficacy trial not feasible, but an immune marker is applicable, comparison of the immune response to a vaccine that demonstrated efficacy would be acceptable. And that's what we are doing, for example, with COVID-19 vaccine. In case we are in a difficult situation in which there is no course of protection and no possibility of bridging immune responses because there are so many uncertainties, then we will need to discuss what could be the alternative strategies. And in any case, whenever we approve a vaccine, particularly if it's based on an immunobridging strategy, it will be very important to conduct post-approval effectiveness studies in order to demonstrate what is the level of effectiveness achieved. Thank you very much.
Thank you, Marco. That was hugely comprehensive and I think very helpful. So thank you. We now open the floor to both Yunda and Marco and invite questions from everyone. So please go ahead and either pose your question or put your hand up, and we will accept any questions that might be out there. So I see a hand up from Elizabeth. Elizabeth, please go ahead.
Sure. So just quick, I'm an immunologist at ViiV Healthcare in translational medicine. And so one of my interests is specifically BNABs. And so my question is for Yunda. I love your study, and I love, the work that you're doing, kind of like what I was talking about with looking at the tails of the data. And I was curious if you guys have any plans to or are looking at, do the individuals that have the higher, PT80, do they end up with any sort of vaccinal effect caused by the BNAB administration? So are you seeing any increases or creation of an anti-HIV CD4 or CD8 T cell response, or are you seeing any sort of changes in immune markers like pro-inflammatory cytokine changes that could suggest that there's a vaccinal effect when these individuals, presumably they're being exposed, and each time you're exposed, you may have increased antigen presentation and possibly immune responses. And so are you looking at those at all? Do you plan to?
Yeah, that's an excellent question, Elizabeth. The answer is yes and no. We did look at the anti-BRCA1 anti-drug activity, immune responses, in our earlier phase 1 trial, as well as in our AMP trial participants. We recently actually completed a relatively large assessment of anti-ADA antibody levels in our AMP trial, about 400 AMP trial participants, including those who had reported, BRCA1 infusion reactions, including those who during the study remain uninfected or those who acquired HIV infection. Overall, we have seen very, very low, less than 8% of participants who had developed any level of ADA across the entire course of the trial, 80 weeks, after 10 infusions. But the no answer part is that we haven't looked more in detail in terms of inflammatory markers, other cytokine level induced by these BNAB infusions. But overall, from the binding level, from the neutralization level, in terms of ADA, we've seen very, very low responses.
Mm-hmm. Yeah, I think I'm very curious about the possibility that the neutralizing antibody, although it's neutralizing, it could also form immune complexes and be eaten up by dendritic cells or monocytes, macrophages, and present that antigen to create a T cell response. So have you looked at T cell responses?
Not
No?
T cell responses.
Okay, thank you.
So, Marco, maybe I can pose a question to you. Just we started out with a talk about the importance of combining more than one biomarker in terms of using these as correlates of protection in a regulatory space. What is the feeling about that in terms of, and I think it alludes a little bit to your comment about generalizability. So, how can you be sure that this can go beyond the population which it was studied, etc.? So how does the regulator approach that?
Yeah, I think on the question of a composite surrogate marker, I think we have always been open to discuss that, but I have to admit that we saw the complexity. And for example, we discussed this for influenza or Ebola also, which Ebola is a very complex disease and very difficult to find a course of protection. And we have not seen any chances of coming up with something meaningful, which is a little bit frustrating, because if you think about Ebola, we know very well that T-cell responses are extremely important. But when we put together the data and try to understand what was the impact, it was not changing the ability of antibodies to be predictive of protection. So I think we are not yet there to have a measurement of T-cell responses in a quantitative way that could lead us to use them as a course of protection. We hope that it will be better in the future. And of course, we are very open to discuss with anybody that has any good idea and wants to engage with us as regulators in this sense. On your second question, yes, it's true that has to be discussed across population. Again, I'm not particularly concerned about the population. I'm more concerned about the vaccine platform, because it's true that, you know, vaccine platform may have huge differences in terms of what you can achieve and the type of immune response. And I think for COVID, the beauty here that it was able to put together studies done with different type of vaccine, different platform technology, and, you know, the same kind of relationship stays, which really indicates the neutralizing antibodies, no matter how they were formed and based on what platform technology, are really correlating to protection. And this is very important. And that's why we could use them in this open way, in which we could have comparison also across different platform technology. But it's not the case for many other pathogens. So we have to be extremely careful here. And it's true that this could be a problem for certain new vaccines.
Great, thank you. Are there any other questions we can put to Yonda or Marco?
I do have one.
Thanks, Deborah.
Yeah, and I don't quite know exactly how to answer this, but and it's directed to Yonda. When you draw these prevention efficacy curves, how and they're based essentially on the placebos observed in the AMP trial, I believe. I'm interested in to what, I don't know exactly how to answer this question, but when you're assessing, you know, the efficacy, the AMP trial overall saw 20-ish% efficacy against the viruses that were circulating, and you're predicting that a triple monoclonal antibody will have 90% efficacy. To what extent do you feel there's I mean, you described some of the things, some of the extrapolation that you're doing to different regions of the virus, and you sort of, because you've only got data from BRCA1. But I actually wanted to understand from the AMP data, to what extent is the efficacy you're projecting, you know, how comfortable are you about the support of the AMP data for the modeling, for the efficacy you're modeling? I don't, hopefully you can sort of perceive the question I'm trying to ask. I don't know. I just wondered, like, you know, it's based on models. The models are based on lab data. You know, you're putting together all these different pieces. How much uncertainty, how much data support do you think there is from AMP for this potential for 90% efficacy based on the newer, longer acting triple monoclonal antibodies?
Yeah, thanks, Deborah, for the question. I think it resonates very well in terms of the open questions I would like the group to discuss. Regarding how comfortable, like, I personally feel about the prevention curve that we derive from the AMP data. Yes, you're right. You know, we use the placebo data in the AMP trial to derive that curve, because for even placebo recipients, we had participants who acquired HIV during the study. So we isolated their virus and measured their IC80, the VRC01 IC80 versus their acquired virus from the placebo recipient, so that we can draw that curve. And then it's if it on its own, I think we do need to be cautious and on this. But at the same time, as I also presented, we observed very similar curve in the non-human primate studies that included, you know, over 250 animals that studied 16 different BNAbs targeting different sites of the HIV trimer. So, and then the added evidence, I think, gives us more comfort, more confidence in terms of, you know, how well the data being captured in our perception. But in terms of your question about how comfortable we feel regarding inferring whether the same kind of PE versus PD80 relationship can be transported to another BNAB or even a BNAB combination? We don't know. We don't know. But based on the data, I do feel quite optimistic, given that the VRSO1, as much as that's one of the best BNAB that we had available at the time where we launched AMP trial, but now years have gone by and the science and technology has really advanced. And nowadays we have these engineered BNABs that have been so much more potent. I was like wowed when I was seeing the data. They were like 10 times more potent neutralization potency than what we observed in VRCO1 or in the first generation, early generation BNAB. What that really means is, in order to obtain the same level of PD80, you only need 1/10 of the concentration of the drug amount. So like in VRCO1, in AMP, you need a 30 mg per kilo. But because the neutralization potency is 10 times more, literally you just need 3 mg per kilo in order to achieve the same PD80 against the same virus. So that's one part. So now we are looking at these much more neutralization potent BNABs. Also, the half-lives of these BNABs have been dramatically increased. The half-life of VRCO1 is around 2 weeks, 15 days. But the half-life we've seen with the LS mutation, with the matrix optimized BNABs, they had these half-lives like 10 weeks, 5-6 times longer. So that's just meaning that not only you need much lower concentration to obtain the same level of PD80 against the same virus, but you are able to maintain the concentration of the BNAB at a lower dose for a much longer time. So that's why we're really in a position to talk about longer infusion interval. So in AMP, we had every 8 weeks because the half-life was 2 weeks. But now we're really looking at, 6 month, infusion interval for these newer generation BNABs. I personally feel pretty optimistic. And we have also seen in vitro data, and not just from our analysis, from, Betty Colbert, from others, in terms of when you put BNABs that are complementary to each other, you put them together, you can see really the neutralization activity is acting complementarily in neutralizing the virus. So really, with a combination targeting different epitopes on the trimer, I think you're really seeing this synergistic kind of action in neutralization. At least in vitro, we see that very clearly. And, we've seen the papers that I cited on my slides, in vitro data, and we feel pretty comfortable. You can see the line kind of gets on the identity line very well. We can pretty comfortably predict these neutralization titer based on the in vivo IC80 of a single BNAB combination, as long as you know the concentration. And for the concentration of these BNABs, these are monoclonal antibodies. So unlike the small molecule, drugs, the pharmacokinetics actually is pretty predictable. I think we're pretty well they're mostly pretty well understood in terms of the trajectory of the concentrations over time. So overall, I think all these pieces adding together, personally, I feel very optimistic about this combination BNABs. Of course, we need data in a real trial to test these hypotheses.
Great. Thanks, Yunda. And I like that little injection of optimism into the webinar. We kind of needed it, I think. I'm also intrigued, so I hope you stick around. Maybe we'll be able to come back, the role that NHP is playing in this regard as well. So I think also injecting a bit of optimism about the animal models in this setting. So thank you to both speakers. We appreciate that very much. And please don't leave us. We are moving now, though, into the discussion part of the webinar. And I'm going to just raise sort of the kinds of questions that might be going through our minds. For example, the practical use of biomarkers in efficacy clinical trials. This question of one or more being identified. How would the markers be included in the clinical trial protocol? What about monitoring them? What else would be needed to ensure appropriate power? Would we still need to measure efficacy by other means? What would be the response of trial participants to the use of these biomarkers? And of course, at the end of the day, the regulatory hurdles, some of which I think we heard very clearly from Marco. So with that, it's my pleasure now to introduce the discussants, and this will help us really think about biomarkers in the setting of clinical trials. So here I'm going to introduce first my good friend and colleague, Mike Cohen from the University of North Carolina at Chapel Hill. And then we'll hear from Pali Das, who is at Gilead Sciences, also wonderful colleague. And then finally, welcome Daisy Oya from AVAC, who will be giving a very important other perspective. So let's start first with Mike, and then we'll work through the rest indeed.
Thanks both to Dr. Cavalieri and Yunda for their talks. Just briefly, with respect to the AMP study and BNABs and kind of the way looking forward, I think first from Barris, Yunda, and Peter, the work they did was called by one of the reviewers in their submission brilliant. And I think really it was extremely helpful to the whole HIV field to undertake that. And it's both inspiring and daunting because the concentrations of antibodies that are necessary are probably beyond what we know how to do with any vaccine currently. So it kind of then shines a light on the idea, well, the monoclonals in and of themselves are PrEP agents and could become PrEP agents if they were cheap enough and effective enough. The problem right now we have, and I wouldn't call it a problem, I'll call it an opportunity, is we have certain numbers of antibodies available from Rockefeller, NIH, and a few other places, but not a huge number for HIV. And our thoughts are to kind of go where Yunda ended her comments, which is she's predicted, she and others have predicted based on in vitro data, that if we give these 3 drugs to high-risk people, we will prevent HIV readily. Either that's correct or not correct. And I think that we'll be in a position to test that. But if it doesn't work perfectly, I would leave open the door to the notion that monoclonals are being developed very rapidly in infectious diseases at a rate that was not going on 5 years ago. Monoclonal antibodies have been popular in rheumatology and cancer and dermatology and other fields for many years. Infectious disease specialists have all focused on small molecules. COVID brought us to rethink this, really. And that brings me to kind of a soapbox. And with respect to the regulators, we think it's extremely urgent to create an environment where monoclonal antibodies for people who cannot respond to COVID vaccines can be approved based on their in vitro activity and or neutralizing titer activity. And we're working pretty hard at kind of following Peter and Yunda's work with Dean Fullman in trying to kind of organize our thinking about how this might work out. Because there's 10 million people in the United States alone who cannot receive protection from vaccination for SARS-CoV-2, and the only option would right now their only option is the Evusheld combination, and the variants of concern create trauma for that combination or lack of efficacy. And yet we know many other monoclonals are in the wings, and so in summary, for the regulatory activities, I think it was stated quite well that we can't really identify the T cell contribution, but we can better and better understand the antibody contribution. And in parallel, we can understand if we're going to give passive infusion, what concentrations of antibodies might let regulators move forward without further clinical trials, which is our really highest priority right now in that space. So I'm gonna not try and talk. There's a lot of other smart people on this call, but I think the issues you laid out are really important. And there's an urgent problem I have for HIV because of my friends in the small molecule business. There might be a little less urgency than there is in the COVID business. And then in the RSV business, which is devastating the United States. And then what I think we'll see is lots and lots of diseases that have never been approached by small molecules will suddenly become accessible to passive immunity, either for treatment or prevention. And I think we have to be open about that and create a space for that to occur. So I'll stop. Thank you, Linda, again, for letting me speak.
Thanks, Mike. And obviously with that, lots of conversations about how we make those then available, cheap, accessible, etc. at the same time. But perhaps a subject for another day. My pleasure now, Mapali, to ask you to give your intervention. Thank you.
Well, first of all, thank you for inviting me to speak today. And I feel quite non-prepared to do so because I'm not a vaccine trialist or expert. And I was thinking about the questions that you've raised, and there's so many parallels to small molecules in HIV prevention and challenges that we've had in preventive clinical trial design that I thought, if it would be appropriate, that I would speak about those parallels and the similarities and what lessons can be learned and used. So if we think about the perfect potential cocktail of antibodies that might work for HIV prevention, and we're able to figure that out and replicate those antibodies and deliver them, then we could look at the concentrations and sort of the exposure, if you will, like we do looking at exposure of small molecules. And once we've determined the range or the appropriate concentrations, we could make a argument of extrapolation. And this is similar to an approach that we'd like to use in HIV prevention trials. Once we've established, for example, the exposure range, the threshold for efficacy for HIV prevention, and the range, you know, between IQ of whatever and your maximum range that we know is the efficacious and safe range for that drug, then hopefully we won't have to continue to do fully powered 5,000, 10,000 person trials to evaluate maybe the efficacy in a different population, that we can't recruit the same amount of folks that we could for major pivotal trials. And maybe in the future, we could use a combination of that exposure range that we've determined for efficacy. And for us in HIV prevention, we're very lucky that if it works in treatment, it's usually historically worked in prevention. So we have an idea of the treatment range of exposure efficacy. We'll know what the prep range of exposure efficacy and we could extrapolate from that if we can achieve safety and that similar exposure range in a different population, that we could extrapolate efficacy. And maybe in the future, we could do a middle step of showing, or an earlier step of showing, efficacy in NHP, efficacy in some kind of in vitro and lab determined environment, and the combination of the appropriate concentrations, and then extrapolate that and have that be the next stage of design, maybe for HIV prevention trials. And I think that we could extrapolate that also to vaccines, looking at those biologic markers, making sure they're in the appropriate, whatever the correct term is in terms of concentration or exposure range, and extrapolating. But I do think that at some, before we get to that stage for preventative vaccines, we will have to demonstrate efficacy in a real human population prior to moving forward. But I thought I would share that perspective from what I do know about, rather than talking to all of you experts about something I don't know about. So I hope that's helpful, Linda Gale.
Thanks, Mapali. It was indeed. And, I think what you've also introduced, which I think is quite intriguing, is because we've talked about how can NHP, how can in vivo help, and here you introduce the sort of infected positive population, and what's the role potentially there in helping us sort this out. And I think that is quite interesting. Typically the prevention and the therapeutic vaccine worlds have been quite siloed. We are seeing more integration with cure and I think Mike's call to bring the monoclonal antibody world and active immunization closer together makes a lot of sense. And maybe, it is time to say, what can we learn from each of these worlds? But I absolutely loved your analogy. I think that did make things very clear. So thank you for that. And we're just going to have our third intervention before we move into our discussion. And Daisy, over to you.
Thank you so much, Linda Gail. I hope you can hear me. Yeah.
Perfect.
Yeah, so I'm speaking as a prevention advocate, and I want to say that I'm very excited with this innovation, especially that the biomarker agenda is now re-energized following, this work that happened, around the AMP studies and to discover this PT80 biomarker. So this is a very important first step. And as Mike said just earlier, it can transcend not only HIV, but also, other disease areas if we get it right. Of course, there are very, very many challenges ahead before we can get there, and especially for HIV vaccine, for which, we don't have any efficacy results, from any clinical trial. So speaking as a prevention advocate, I wanted to pose a few questions as well as make some recommendations. And the first one is, in terms of a question, is whether the subtype matters of the virus. So of course, the AMP study happened primarily in B and C subtypes, populations with B and C subtypes circulating. And I was wondering, are these results, especially this biomarker that we've discovered, will they apply to all the HIV subtypes? And I think when Dr. Marcos spoke, he pointed out to the need to include diverse populations in this biomarker discovery work, so that it can be more broadly acceptable and the results that we find can be more acceptable. My other question is whether there's any research happening to collect to discover biomarkers that maybe are read out of easier to collect samples, because currently AMP is studying the blood and so on. Can we get biomarkers from saliva, from urine, from hair samples, which are easier to collect, especially in resource limited settings? On this same point, it is clear that it will be necessary to collect biomarkers, to collect samples and process them, much more aggressively as we, ramp up this biomarker discovery work. And I just wanted to point out to the UNAIDS ethical guidance, point number 9 on informed consent, which, recommends that data banking and biobanking processes should be in line with international and national legal and ethical guidance documents. So that will need researchers to consent participants in terms of what is going to happen to their samples. And I'm sure this is something that the HPT and other researchers, including Gilead with their purpose studies, are considering. Also, from a community perspective, I wanted to point out some of the cultural barriers that might arise when samples are collected, and particular samples. People have there are certain taboos against collecting certain samples, and so this is just something to take into account and work around. Also the need to sensitize regulators and ethicists in our continent here in Africa and elsewhere, early on, before they are confronted with protocols that are looking to use biomarkers as perhaps surrogate endpoints in future trials. So this will help with the protocol review as well as potential approval of new drugs, new vaccines in the future. The other point is that we need a very careful research literacy for advocates, and this is what ABAC has been doing, and we are committed to this. in the recent past, we actually did engage very closely with various groups who were using recency assays as a surrogate marker for early HIV infections, and we asked very many questions. But of course, by the time we were engaging, the ship had already sailed, and some of our questions were not really answered. Whether so we want to do this earlier. In other words. In closing, I would like to say that even as this work gathers pace, we would advocate that it doesn't overshadow existing efficacy trial designs, including comparator trials such as the ones that are being used in the Purpose program and other phase 2 trials, as well as the new experimental medicine designs. And finally, most importantly, that HIV prevention using available options, including condoms, oral PrEP, ring, injectable PrEP, treatment as prevention, post-exposure prophylaxis, PEP, as well as also structural interventions. We hope that, and we will continue to advocate that this continues to be supported and funded and rolled out with equity and with an increasing sense of urgency, because people need prevention right now. And we hope that this can continue, with a renewed sense of urgency. Thank you.
Thank you, Daisy. Well said. And, couldn't agree more that we need to get better at this. I always say, it's walking and chewing at the same time. So not only getting out the prevention we've got, but obviously also really investing in the fact that we do need a prophylactic vaccine that is affordable and effective, and we need it urgently. So it really is about doing all of the above. So great. Thank you so much for those wonderful remarks. I'm going to ask all of the panelists, including, you're welcome, Marco and Yunda, also to join, just in terms of answering some of those questions that Daisy has posed, but also throwing it open to the broader group to say, please bring your questions or remarks or comments. We'd love to hear them and continue the discussion now. So pose them in the chat or feel free to unmute yourself and speak. And maybe just to kick it off, Daisy, I'll ask you, I was struck during, and I don't know how other people found this, the experience of the COVID-19 sort of recent experience, just in terms of community and the understanding of the biomarker of antibodies. I felt like it was almost, a sort of a rush to, and in some ways, sometimes I think a misunderstanding is, as Marco has pointed out, the importance of T cells, yet this huge fascination with antibodies. Do you want to just talk about community understanding and that literacy around biomarkers and what might need to be done as we go forward?
Linda Gale, is it addressed to me?
Yes, Daisy. Yes, to you.
Yeah. I think the very word biomarker is already a jargon in terms of community understanding. So I think that it needs like some serious translation before you can, you have to use maybe things that are relatable before you drop the biomarker word. I do not think that at least in my experience that communities anywhere understood the idea of biomarkers, and that perhaps some of these vaccines were being evaluated based on, biomarkers as correlates of infection. I think what we saw mostly was that these were trials that were studying this population with the vaccine versus this other population without the vaccine. So I would, at least from my little understanding of, of the COVID vaccine space, I do not believe that people really understood that there was biomarker research, biomarker-related research that was happening around COVID. Thank you.
Great. No, thanks, Daisy. So, maybe again, a call to action that we have a lot of work to do in terms of literacy and so on. Mapali, picking that up, I know you are, the purpose study is using a counterfactual. What is the experience on the ground, just in terms of, Jim raised the sort of other types of counterfactuals that may be out there. Do you want to just reflect on that for a moment?
Yeah, thank you for the question. Yeah, so, we worked very closely with the Forum for Collaborative Research and many folks on this call to implement Jeff Murray's proposed counterfactual design in the Purpose program. So both from a perspective of educating, really involves educating a wide variety of stakeholders, including regulators who may not be aware of the design or feel like we must use the gold standard RCT, non-inferiority design, when you have an proven efficacious method already. But so regulators, other investigators, scientists, providers, physicians, and site staff, and of course, people who are advocates and leaders for the potential participants we seek to recruit. So when we launched, even before we launched the trial, we did 2 years of engagement processes, co-sponsored with AVAC, on our own, meeting with everyone, making sure that people understood the trial design and would feel comfortable explaining it to their participants, to each other in the community, to colleagues about that trial design. And with respect to the actual science of the trial design, it's a commonly accepted pathway for oncology products and hormonal or any kind of contraceptive product. Because, and I was just talking about this morning, let's say you have 100 women and it's unethical to randomize them to pregnancy prevention or not, to birth control or not. So the way they work on evaluating new pregnancy prevention options in regulatory agencies and for pivotal trials is they use something called the Pearl Index. So you look at the expected rate of pregnancy in 100 fertile women, which is 80 per 100 person years. You put those same 100 or whatever N you need for safety, let's say 300 women, and you look at how many pregnancies there are, how many failures of the contraceptive. And if you have 1 to 2 per 100 person years, you can be reassured that you've had a 40-fold and 80-fold reduction in pregnancy and that your contraceptive is working. So I always use that to explain what we're now trying to do in HIV prevention, which would be suitable for HIV vaccines as well, which would be you establish your counterfactual or your background HIV incidence in the population you're screening for the study. So our study designs have a screening incidence cohort where everyone is tested for HIV. And then we use a recency assay to determine if the person has been recently infected with HIV and use that to back calculate HIV incidence with many of the colleagues on the call and their institutes helping advise us on how to do so. And then we have that background HIV incidence. And then we're able to compare the number of infections in our active arm with the expected HIV incidence for that amount of person years. And that's what we're doing in Purpose 1 and Purpose 2. The beauty of this design is you can evaluate 2 products at the same time. So for Purpose 1, we're looking both at lenacapavir and at FTC TAF or Descovy in cisgender women. And then we use an active control of Truvada to reassure people, for people who are familiar with how FTC TDF works for prevention. So those are the trial designs in Purpose 1 and Purpose 2 using that counterfactual. And I think there are many challenges and pros and cons of using recency. One thing that we will do is also use other methods to reassure that the recency assay was performing well and in the diverse populations we're recruiting for the trial, including surveillance data and recently conducted vaccine or other trial data that tells us the HIV incidence in the similar cities and geographies where we're conducting the study.
Thanks, Mapili. Thanks. I think that's helpful. So, Mike, HPTN must have thought about this. How are we going to design the trial of the new monoclonals using PT80? How does it come together?
Well, this whole trial design thing that Mopely got into is a consensus building exercise that I think you're obviously part of, right? And Deborah Donnell, she's still on the call, is part of. For most of the things we're trying to do, well, for HIV, we probably have the luxury of doing randomized controlled trials to prevent incidents, but the ethics that surround those trials are complicated, and that's how some of the designs, such as Mobley described, surface. But for monoclonal antibodies for diseases for which there is some sort of either the incidence of the disease is too low. Monoclonals are the best example in some ways, because those vaccines, while we wanted to study prevention of infection, that was never really studied. What we studied was prevention of progression of disease. So because we studied prevention of progression of disease, and because this is the VOCs evolve, it creates a big problem for ongoing vaccine trials, which brings us to the immunobridging that Dr. Cavallari so nicely described. But for monoclonals, I don't think we can do clinical trials because the incidence is too low. The people are preventing, again, I'm focused on compromised hosts who need SARS-CoV-2 monoclonals. They're protecting themselves in so many ways. They're vaccinated. So there we have this desperate need for surrogates. And so there's some spaces we can do clinical trials, and some spaces we need some sort of surrogate that a regulatory agency is comfortable with, that opens up a field. Now, I don't want to be too long-winded. There are the following kind of designs: an RCT of some sort or the other, but it gets into a lot of ethical issues of what is the comparator. If you're living in a poor country and the comparator is the worst possible drug, why don't you have the best possible comparator? Because the various companies are working on the country. We've already heard about the counterfactual design, and that's quite important, and U.S. regulators have agreed to that design. There's the condom refuser design, right? Only enroll people who say they're not going to do anything anyhow in any trial. So you're convincing people who say they're going to do nothing to do something, and then the placebo group are people who said they're going to do nothing anyhow, but every time they come back, you offer them something again, right? And hope that all of them don't end up on something, which is a danger in the trial. And lastly, there's a design that we've talked about a lot, which is the kind of futility design. And I'm not opposed to this. We take the best agent we have for PrEP, let's say, and then we take 3 monoclonals and we enroll very highest risk patients, and we stop the study if there are too many incident cases in the comparator, right? So instead of a giant trial, we end up with like an emergent futility event if the 3 drugs don't work as well as you. And the end of this is not meant to offend your optimism. I have the same optimism, but our concern about that design is it would create a negative situation for further drug development. If we do that design, but we're anticipating more monoclonals and we're open to that design, then I think it opens the space. If we close this forever through that design, that's a disaster. And there's the ethics that surround that design. So sorry to be long-winded. I'll stop. You asked a question. Can I answer one other thing about the clades? Coming back to Daisy's question.
Yes,
Yeah, the clade, that is a really big deal. That's the same as variants of concern, right? And so there is no reason to believe that clade A, some clade, it's rare, is going to be susceptible to the 3 monoclonals that Yunda described. And we know for sure there are differences in clade C and B across the monoclonals. We chose the ones we chose because they're very active against clade C. We worked in Africa because they're very active against clade C, but and they also work against clade B. But there are some other BNAbs, it will not work against clade C. And then our South African colleagues have CAP 256, which is very specific to clade C. So your question is very well taken, and it does really make a difference what clade you're working on, exactly as you asked.
Thanks, Mike. Thank you for answering that. And we're out of time, but I'm going to actually give Peter Gilbert the last word here, just in terms of he's posted in the chat. And intriguing, yet vaccine efficacy trials could do the same kind of virus-based neutralization sieve analysis that was done in AMP, a powerful way to learn about the correlates that perhaps is underutilized in vaccine efficacy trials. I don't know, Peter, if you can sort of sum this up in 1 minute. It's a but or leave it out as a tantalizing sort of concept that maybe we can pick up in a future webinar, but please go ahead.
Yeah. Well, thanks, Linda Gale. Yes. So in the AMP correlate study, we really couldn't study the PT80 directly as a correlate because we couldn't determine the acquisition time clearly enough. So the empirical data from the study was that prevention efficacy was better against viruses that were measured to be more sensitive to neutralization by VRC01, a concept David Montefiore for the neutralization sieve analysis. Perhaps that concept has been underutilized for vaccine research because one could characterize viruses in persons with clinical endpoints in vaccine efficacy trials for the vaccinees versus the placebos, or for the vaccinees versus a control arm, and apply different assays, neutralization, other kind of effector function assays, and use this technique as a foil for determining which assays for which you can detect that basically the breakthrough viruses vaccines are more resistant to the vaccinee sera with a given type of immune response compared to the placebos who have the case. So yeah, I just wanted to put that thought out there that this might be an area for more attention.
Great. Well, thank you. Thanks, Peter, for that. Thank you to all of our panelists, all of our speakers in both sessions. And I'll invite Kerry back to join me just in saying thank you and good night to everyone. I want to remind everyone that the guest presentations have been recorded. The recordings of the presentations will be made available by the team represented here. Of course, I want to thank the IAS staff for all of their hard work in putting this together. There's a great team who conceptualize and put this all together, so thank you to them. And of course, there will be a report as well, which we'll make sure is shared with all. And with that, picking up on Peter's point, I think there's work to be done. We heard that there are real gaps around T-cell immunity, gaps around the NHP model, gaps around how community understand, gaps how we design trials, how we put these various silos together to see how we can really put things together. And with that, I'm going to hand over to Carrie to close us down. Thanks, Carrie.
Great, thank you, Linda Gale, and thank you to all the speakers and participants, and also the IAS, Tuli and Roger and others who helped organize this webinar. I think this is a really important conversation to have. I think this is a critical issue, for people living with HIV and people at high risk for getting HIV infection. And this is a great first step, and I look forward to carrying on this conversation in the future with this group and others. So thank you.
Thanks all. Good night.
Bye-bye.
Thank you, everyone.
Bye.